Welcome back, everyone. Dr. Linda here. In the next short video, we are going to continue with muscle physiology, and we need to discuss the three different types of muscle proteins. The three different types of muscle proteins are contractile proteins, regulatory proteins, and structural proteins. The important contractile proteins are actin and myosin. Those are the two biggies. Actin is the thin one, and myosin is thicker. So myosin is this one here. Actin is this one here. Now, one of the things that I want to point out to you is that myosin has something called myosin heads. And myosin heads have a really important job, and they have these binding sites. They're called actin binding sites. So somehow this structure here has to make contact somewhere there on actin. Okay, so this is actin here. And actin has some regulatory proteins. One is called troponin, and the other is called tropomyosin. Okay, let me see if I can erase a little bit here. Okay. So now we're looking at the regulatory proteins on actin. Right there is actin. Troponin are these little blue staples that we see here, and these staples are holding something in place. They're holding tropomyosin in place. Tropomyosin are these ropes. You see these like strings that kind of go like this, right? And there's another one on the top that kind of makes another spiral like that. And tropomyosin is covering these binding sites. We see here myosin binding sites that are covered by tropomyosin. Now, if the tropomyosin was not covering the binding sites, then the binding sites would be exposed. If the binding sites on actin are exposed, guess what these myosin heads that have actin binding sites do? Well, the binding site is going to find the binding site on actin. They're going to make contact, and they're involved with the contraction. But in this particular case, they're covered because troponin, these little staples, are holding tropomyosin in place. So we need a staple remover to pull out troponin. And guess what that staple remover is? Calcium. So if calcium was around and available, it's going to pull out troponin so that the tropomyosin, these ropes, can shift or rotate and move out of the way. When they shift and rotate, the binding sites become exposed, and then muscle contraction can take place. These structural proteins, titin, nebulin, alpha-actin, myosin, and dystrophin, these are structural proteins that give the shape to the muscle, some of their tension to the muscle, their tone to the muscle. We've known that if there's an autoimmune condition where the body attacks this type of structural protein, we call it muscular dystrophy, right? So those structural proteins deal with the alignment of the muscle. They deal with the stabilization, the elasticity, and extensibility of it. The contractile proteins are going to be actin and myosin, and the regulatory proteins, that's the on and the off switch, that's troponin and tropomyosin. Troponin I refer to as the staple, and tropomyosin is the string or that rope that covers the binding sites. And then Calcium at the bottom here, calcium is the key or the staple remover that removes troponin so that the tropomyosin shifts out of the way. So tropomyosin is going to extend the entire length of actin and it covers the binding sites. Troponin holds, it's the staple, it holds the tropomyosin over the binding site. Okay, now actin is made up of F and G proteins. Okay, F and G proteins, I'll show you what those are. Each little sphere, like this is a G protein. All of these are G proteins. And remember, you're going to have that tropomyosin that covers them, right? Like that. Um, the F, the F strand is when you have all those G molecules combined together. So here's a close-up look. This is actin. Here's actin. These are binding sites. We see those like purple little targets in the center. Those are binding sites. But notice how troponin, the orange rope, is covering the binding sites. And here's troponin, the staple that's holding tropomyosin in place. Now, if calcium comes in, calcium is going to attack the troponin. It's going to remove it so that the tropomyosin can shift. And when it does, these myosin heads can actually attach to them. Look at all the myosin heads, right? It's not just one. There's so many of these myosin heads, these little hands or fingers that can attach. So think of it as on the bottom right, we have a myosin tail, and then we have these hinges, right? Here's a hinge. Here's another hinge. And I like to refer to this as like, think of this as the hand, like your wrist or your hand with your fingers. And then, so let's say this is your hand with your fingers. This would be your wrist joint, and this would be your elbow joint. So your elbow can bend here, and then your wrist can bend here. That's important. I want you to keep that in mind. Because when the myosin makes contact with actin, first it bends at this elbow joint, right? Let's remove this here. It's first going, when the binding site is exposed on actin, that bends first so that this whole thing moves up and makes contact. And then it bends here at the wrist and creates this little flick. And that's what creates a contraction. Look up at top, you can see again you have these G proteins. Combined, you get an F strand. And notice that we have, in this case, these yellow circles are the binding sites. And again, they're covered by tropomyosin, and we have troponin holding it in place. When the myosin heads, all of these, make contact with actin, they create a contraction. So if you look at the H zone here and the H zone here, look how much smaller the space is between this actin and this actin. We call it an H zone. It's much smaller because it contracted. There was a sliding filament that took place. When actin and myosin meets, it's going to slide the actins closer to one another. So the H zone becomes much more narrow, and the H zone is just the distance between this actin and this actin. Okay, so what's taking place? Let's review again. Here's the motor end plate, right here. This is the motor end plate. When I say motor end plate, that's the muscle. Here's the synaptic terminal, which is at the end of the axon, and we can see the neurotransmitters here that have acetylcholine. Here's the transverse tubule, the invagination. Okay. Somehow we're going to see calcium. Calcium is going to come in. It's going to tell the neurotransmitter to release through exocytosis. So you're going to have, number one, acetylcholine is going to be released. It's going to bind to the receptor. Those receptors are on the membrane of the muscle. Excitation is going to happen. Number two, the action potential goes down the T tubule. Right here is the T tubule butting up against the sarcoplasmic reticulum. What does the sarcoplasmic reticulum store? Calcium. That calcium, there it is, it's going to be released. And now in your brain, in your mind, we know what actin has. Actin has those regulatory proteins, troponin and tropomyosin. If calcium comes in there and goes after troponin so that tropomyosin shifts, then the binding site becomes exposed. So hinge, number one, bent, and it allowed the contact right there of myosin to the binding site on actin. Eventually, at hinge number two, it's going to flick. It's going to flick and create this sliding filament to create a contraction. All right, so these steps, 1, 2, 3, 4, and 5 on the left are going to be found on this page, 1, 2, 3, 4, 5. Let's go back here. The steps that end a contraction. Well, if acetylcholine was responsible, then we need to remove the acetylcholine by using an enzyme called acetylcholine esterase. Acetylcholine esterase, anything that ends in ASE, is an enzyme. Remember, if something ends in an OSE, it's a sugar. So don't confuse ose, like sucrose, fructose, glucose, with an ACE. ACEs are enzymes. So acetylcholine esterase is an enzyme that's going to break down acetylcholine. And when it does, if there's no acetylcholine, then we don't have that excitation taking place. If there's no excitation going down the T tubule, then nothing is going to stimulate the sarcoplasmic reticulum. So what's going to happen to all that calcium? It's going to go back in here for storage. So the SR, the sarcoplasmic reticulum, is going to recapture the calcium. And if there's no calcium that's stimulating the regulatory protein troponin, then the troponin tropomyosin complex go back to their normal resting state. The binding sites on actin are covered, and then the myosin heads go back to their resting state, and the contraction ends. All of those steps are stated here. So at the neuromuscular junction, acetylcholine is released. When acetylcholine is released, it's going to bind to the receptors, those sodium channels, remember? It's going to create this action potential that spreads down and goes right into the invagination of the T tubule. The T tubule butts up against the sarcoplasmic reticulum that's going to release the calcium that it stores. That calcium is now going to bind to troponin, remove that so that the tropomyosin complex shifts, and it's going to expose the binding sites on actin so that a cross bridge can form. The cross bridge is that union between the myosin head and the binding site on actin, so that a contraction begins as you have all of those myosin heads that are going to attach, and then they pivot. That flicking of the myosin head, they call pivoting. And then it can detach. But that detachment requires energy in the form of ATP. For those myosin heads to detach, it takes ATP to do that. To end a contraction, the acetylcholine has to be broken down by acetylcholine esterase. There's the enzyme, acetylcholine esterase. So the sarcoplasmic reticulum reabsorbs the calcium. And when the calcium concentrations approach their normal resting level, The tropin and tropomyosin complex return to its normal resting state. There's no more cross-bridge interaction, so the sliding filament theory doesn't take place, and then the muscle relaxes, returning it to its resting length. What are some of the events that can influence that NMJ to produce the action potential? Well, we said that the voltage-gated calcium channels in the neuron synaptic end bulb open. And that's when you have the influx of calcium. That influx of calcium tells that vessel that contains the neurotransmitter at the synaptic end bulb to release acetylcholine through that method called exocytosis into the synaptic cleft. Right? The neurotransmitter binds to the sodium channel on the motor end plate, and it causes an influx of sodium into the muscle. That's what creates the action potential or the depolarization of it. How do you break down the neurotransmitter? NT is neurotransmitter. It gets broken down by acetylcholine esterase. That is the enzyme. Okay, when we come back, we'll get into the contraction and relaxation of the skeletal muscle once again. To review.